I welcome all of you to this session of the SIMG Gene Talk webinar series. I'm Dr. Prajnya Ranganath from the Department of Medical Genetics at the Nizam's Institute of Medical Sciences, Hyderabad. I'll be talking about the structure of the gene and the genome and genetic variations. This would be the outline of my talk. I'll be talking about the organization of the human genome first, followed by normal gene structure, then types of genetic variations and zygosity. After that, I'll give you some examples of common genetic diseases associated with different mutations. Now, coming to the organization of the human genome, as we all know, the human genome is made up of around 3 billion base pairs. Most of this is arranged within 46 chromosomes inside the nucleus, and this is referred to as the nuclear genome. A small fraction of the genome is present within the mitochondria, and this is referred to as the mitochondrial genome. Now, if we look at a comparison between the nuclear genome and the mitochondrial genome, the nuclear genome is made up of about 3,200 megabases. It is linearly arranged within the nucleus as 46 chromosomes. There are 2 copies of the nuclear genome per cell. And on the whole, there are about 19 to 22,000 protein coding genes present within the nuclear genome. Most of the genes have introns, and of the entire nuclear genome, only about 1.5% is actually coding DNA, and variants in the nuclear genome follow the Mendelian patterns of inheritance. Whereas the mitochondrial genome is made up of about 16.6 kilobases, and it is circularly arranged within the mitochondria. There are about 10 to 50,000 copies of mitochondrial genome within each cell, and there are about 37 genes within the mitochondrial genome, of which only 13 are protein coding. Usually, mitochondrial genes do not have introns, and about 93% of the mitochondrial genome has coding DNA, and variants in the mitochondrial genome follow maternal inheritance patterns. Most of the human genome, about 98 to 99%, is made up of non-coding sequences, which is referred to as junk DNA. This could include the retrotransposons, the transposons, the lines and the signs, and other non-coding sequences. And as I mentioned before, only 1.5% of the genome has protein coding sequences, and about 19 to 22,000 protein coding genes are present in the human genome. So now, what do these things mean? So retrotransposons are actually transposable elements, which are also referred to as jumping genes. Now, these are DNA sequences which can change their position within the genome. Retrotransposons, on the other hand, are transposons which duplicate via RNA intermediates, which are reverse transcribed and inserted at new genomic locations. Lines are nothing but long interspersed nuclear elements, which are long, non-long terminal repeat retrotransposons. More than 100,000 copies of lines are present in the genome. And the most common type of line is line 1, which is about 6,000 bases in size. The signs, on the other hand, are the short interspersed nuclear elements, which are about 100 to 700 base size retrotransposons. Around 750,000 copies of these signs are present in the human genome, and the most common signs are the ALU repeats. Coming to the structure of the gene actually has a rather complex structure. If you look at it, there is something called a promoter region. Then there's within the main gene, you have the exons and the introns. And then there are other upstream and downstream elements, which we'll discuss in more detail. So if you look at the entire gene, the nucleotide position where the transcription starts is called the initiation codon. Now, this is where the transcription happens, wherein the DNA is transcribed into the mRNA, the messenger RNA. Then after transcription, the messenger RNA undergoes something called post-transcriptional modification, wherein a 5' capside sequence is added, a poly A tail is added at the 3' end of the sequence, and then the introns are spliced out, and finally the mature mRNA is formed. The mature mRNA in turn gets translated into the protein. Now, the point of transcription where the transcription starts is called the transcription initiation site. The point where the cap sequence is added is called a 5' cap site. The point where the AUG is there, the codon where the translation begins is always the AUG codon, and this is called the initiation codon. And where the translation stops, that is called the termination codon. So the sequence of the mRNA between the start of the mRNA and where the translation begins, that point is called the 5' untranslated region. And the sequence of the mRNA between where the translation stops and where the mRNA sequence ends, that part of it is called the 3' untranslated region. Exons are, of course, the portions of the gene that code for amino acids. These are retained in the mature mRNA following post-transcriptional splicing, whereas introns are the portions of the genes that do not code for amino acids, and these are not retained in the mature mRNA following post-transcriptional splicing. So splicing, as we all know, is the process where the introns are removed and the exons are joined together. This is one of the post-transcriptional modification that happens in the conversion of the initial mRNA into the mature mRNA. And the sites at which this splicing occurs are actually the junctions of the exonic and intronic sequences. These are referred to as the splice junctions or the splice sites. And consensus splice sequences are nothing but fixed sequence of dinucleotides at each end of the intron where the splicing occurs. So the donor splice site sequence is GT, which is at the 5' end of the intron, and the acceptor splice site sequence is AG, which would be at the 3' end of the intron. Moving on to definitions of what we mean by genetic variations and zygosity. A mutation is a permanent alteration in the nucleotide sequence of the genome of an organism. When we talk about a genetic variant or a genetic variation, we are referring to a difference in the DNA sequence of an individual when compared to reference genome sequence. Actually, nowadays, the term mutation is no longer preferred. Rather, the term genetic variant or genetic variation is used, but sometimes these terms are used interchangeably. And when we are talking about a copy number variation, referred to as CNV in short, we are actually referring to a gain or loss in comparison to the reference genome of a large stretch of the genome, which is at least 1,000 bases or larger in size. And the size of the CNV can range anywhere from 1 kilobase to several megabases. How do these mutations occur? Mutations can occur spontaneously or because of DNA damage by environmental agents. Spontaneous mutations most often occur due to chance errors in DNA replication. And environmental agents which cause mutations are known as mutagens. These could include ionizing and ultraviolet radiation, free radicals, and exogenous chemicals. DNA repair mechanisms normally exist to repair this DNA damage and correct the replication errors. But some errors which escape these repair mechanisms get retained as mutations. And if the mutations involve important coding portions of the genome, that can result in disease. Now, when we talk about the zygosity of a variant, what do we mean? So what is a heterozygous variant? So generally, the two copies of a gene in the human genome or in other genomes of other organisms are referred to as alleles. When one allele has a variant, that particular variant is said to be present in a heterozygous form. If both the alleles of that particular gene have the variant, then that variant is supposed to be present in a homozygous form. Sometimes the variants which are present on the two different alleles of the gene, of the same gene, may be at different positions of the same gene. So you see here, there is one variant at one particular position in one allele, and the other variant on the other allele is at a different position of the same gene. So then these two variants are said to be compound heterozygous. And when there is only one copy of a gene present, for example, X-linked genes in males, and that particular single allele has a variant, then that variant is said to be present in a hemizygous form. What do we mean by mosaicism? Mosaicism refers to the presence of more than one genetic line in a single organism derived from a single zygote. So gonosomal mosaicism is where a proportion of the somatic cells as well as the germ cells have the mutation. Somatic mosaicism is where only a proportion of the somatic cells have the mutation. And germline or gonadal mosaicism refers to the presence of the mutation in only a proportion of the gonadal cells. Somatic mosaic mutations are usually seen with segmental overgrowth conditions. For example, Proteus syndrome, where you have somatic mosaicism of the AKT1 gene mutation, and somatic acquired mutations are also seen in malignancies. Moving on to the genetic variations, what are the different types of variations? What are the diseases associated with them? So broadly, you could classify variations as substitutions, deletions, duplications, insertions, inversions, and repeat expansions. When we call a mutation a substitution mutation, what we mean is there is one nucleotide replaced by another nucleotide at that particular position. These mutations were previously referred to as point mutations, but now the preferred term for them is single nucleotide variant or SNV. Now, the type of the SNV can differ based on the position and the effects. So, if the SNVs are present within exons, based on the effect that they have, they could be called missense variants, nonsense variants, or synonymous variants. If the SNV is present at a splice site, it can alter the splicing and then it is referred to as a splice site variant. When the SNV is present in the promoter region of the gene, it can alter the gene expression also. So substitution of one nucleotide by another, which leads to replacement of the amino acid by another, is referred to as a missense variant. For example, here we have this normal DNA sequence. Now, as you can see in the second codon, there is a substitution variant, and this substitution is resulting in replacement of the glutamic acid in the second position by arginine. So this is a missense variant. I'll give you an example of a missense variant. This is a very common example. So there are now here we have a family with a 5-year-old male patient who has presented to us with chronic severe anemia and recurrent joint pains. There is third-degree consanguinity in the parents. One elder sibling is similarly affected. Hemoglobin HPLC of the child shows a hemoglobin S fraction of 72%. So the sickle cell anemia would be the most likely clinical diagnosis here. And we know that sickle cell anemia is caused by a specific missense variant in the HBB gene, which is at the 20th nucleotide position, you can see here. And that results in replacement of the glutamic acid bivalent at the 6th amino acid position. So it's a missense variant, and one of the most common examples of a missense variant causing human disease. So at this position, A was supposed to be there and it is substituted by T. Moving on to what is a synonymous variant. Now, a synonymous variant is one where there is substitution of one nucleotide happening by another, but this does not lead to replacement by another amino acid, because the mutated codon codes for the same amino acid as the original codon. So you can see here, GAG is mutated to GAA. However, both GAG and GAA code for glutamic acid, and therefore there is no change in the amino acid. So this is referred to as a synonymous variant. Okay. Now, synonymous variants, otherwise usually referred to as silent variants, usually are benign because the mutated codon codes for the same amino acid as the original codon and there is no resultant change in the protein sequence. However, there are exceptions to this rule. Sometimes synonymous variants can activate cryptic splice sites, alter splicing and produce different transcripts, which in turn can produce a different protein which can lead to disease. And the best example of a synonymous variant altering splicing and leading to disease is Hutchinson-Gilford progeria. So you can see this child here, she's 13 years old, but you can see the aged appearance. So the child has alopecia, lipodystrophy, a progeroid, early aging appearance, joint contractures, and so on. And this is caused by mutation in the LMNA gene. Now, the sequence chromatogram of the LMNA gene shows that there is a substitution here. Okay. So, but what is happening? Even though there is a substitution there, there is no we do not expect any alteration to happen in the amino acid because the original codon as well as the new codon both code for glycine. Then why is it that this particular variant is causing disease? This is because there is altered splicing here at this site. So what happens is, because of the altered splicing, a cryptic splice site is activated by this particular variant. So even though it is not expected to alter the amino acid, because of a cryptic splice site being uncovered, because of altered splicing happening there, 150 nucleotides, it results in the creation of a new transcript, and that transcript has 150 nucleotides less when compared to the original transcript, and therefore the resulting protein has about 50 amino acids missing there, and that is what causes the abnormal protein and thereby disease. Coming to a nonsense variant, then the substitution of one nucleotide by another leads to replacement of a protein code or amino acid coding codon by a stop codon, that is referred to as a nonsense variant. So you can see here in the second, particular thing, here the UGC was supposed to be there, which was supposed to code for cysteine. However, UGA, because of the mutation, it has become UGA, and this is actually a stop codon. So this is where the protein translation will stop, and this is referred to as a nonsense variant. So I'll just give an example of a family of a child who was diagnosed to have a particular condition due to a nonsense variant. Now this is a 4-year-old female child born to non-consanguineous parents who presented with global developmental delay and intellectual disability. She had seizures and facial dysmorphism. Clinical exome sequencing showed a heterozygous variant in the gene, in the TCF4 gene. And this particular variant is actually, if you look at the consequence of the variant, at that position, that codon was supposed to code for glutamine, but now the altered codon actually is a stop codon. So there is a stop of translation there, and therefore it is referred to as a nonsense variant. And then further variant segregation analysis in the parents confirmed that both the asymptomatic parents were negative for that variant. And so the final diagnosis for this child is that she has TCF4 associated Pitt-Hopkins syndrome, which has an autosomal dominant pattern of inheritance. So you can see the variant here, the substitution happening C to T in the child, and both the parents are normal for that particular position. Both of them have the normal nucleotide there. There is no substitution in them. So this child most likely has the variant in the child is most likely a de novo variant. That is to say, most likely has arisen newly in her, and it is not inherited from either parent. Of course, there are exceptions to this. Sometimes you can have gonadal mosaicism for a variant in one of the parents, and that would not be detected in the testing of the blood DNA of the parents. And that, I'm sure you would have another lecture session on that. Now, coming on to a splice site variant, what do we mean by this? So again, I'll give you an example. So here we have a 3-year-old male child who presented with chronic severe transfusion-dependent anemia. The elder sibling died at 7 years of age with similar features. Hemogram showed microcytic hypochromic anemia with anisopoikilocytosis, and hemoglobin HPLC showed a significantly elevated hemoglobin F fraction, fetal hemoglobin of 93%. So the clinical diagnosis here is beta thalassemia major. And when the child's hemoglobin, beta globin gene sequencing was done, the homozygous variant was found. Now this is a splice variant. This is actually the most common mutation that is associated with beta thalassemia. It's referred to as IVS15G2C. IVS because it is present in the intron, 1 because it is present in intron 1, and 5 because it is present in the 5th position of intron 1, and it is a substitution of G to C. But this is the conventional way of denoting this variant, and this is the recent HGVS nomenclature for this variant. So as you can see here, the substitution has happened from C to G, but actually it is in the splice site. This is where the exon ends, and this is where the intron begins. So it's in the 5th position of the intron, and it is causing disease even though it is not in the coding portion. It is present in the intron, but it is causing alteration of splicing, and therefore it is causing the disease in the individual. So this is an example of, again, one of the most common examples of a splice site variant causing disease in human beings. Inversion is where the a part of a gene or the entire gene is actually inserted into the genome in a reverse orientation. So the most common example of an inversion mutation is the intron 22 inversion mutation, which is seen in the factor VIII gene, and this accounts for about 45 to 50% of patients with severe hemophilia A. Moving on to small insertions and deletions, when only one or a few nucleotides are inserted or deleted, they are called small insertions and small deletions. When they are not in multiples of 3, this can lead to a shift in the reading frame, in which case they are referred to as frame shift mutations. And when the same nucleotide or sequence is inserted, it's called a duplication. So here, as you can see, there is a insertion of only one nucleotide here, and this results in the shift in the reading frame, and all the amino acids downstream to this insertion are different from the original one, and this is an example of an insertion causing frame shift. Whereas here there is an insertion of 3 nucleotides, so one additional codon is inserted and thereby one additional amino acid is inserted, but the rest of the reading frame is intact and all the downstream amino acids are the same as in the original protein. And so this is an example of an insertion without frame shift. Again, deletion of one deletion of one nucleotide alters the reading frame. The nucleotides are regrouped, so the codons are different and the amino acids they code for are different. So this is an example of deletion with frame shift. And here, when 3 nucleotides are deleted, one codon is missing, so that one particular amino acid, which was supposed to be glutamic acid here, is missing, but the rest of the reading frame is intact, and so all the downstream amino acids are intact. So this is an example of deletion without frame shift. So now here we have an example of a patient with a frame shift variant. So this child presented with global developmental delay followed by regression of milestones, had coarse facial features, radiographic features of mild dysostosis changes, MRI findings of some changes in the globus pallidus and the white matter. And when we did the clinical exome sequencing for the child was found to have a 1 base deletion in exon 5 of the FUCA1 gene, which is known to be associated with fucosidosis. And in the child, this particular deletion was present in a homozygous form. So both variants, that particular thing which was supposed to be there is missing. But if you see parents, they are heterozygous carriers. So one allele of the parent, each parent would have the normal, in one allele, that particular nucleotide is present, the G is present. Whereas on the other allele, the G is absent. That is why beyond the point of the deletion, you find overlapping sequences, one corresponding to the normal allele and the other corresponding to the abnormal allele in the heterozygous parents. Whereas in the affected child, because there is a homozygous deletion, in both alleles the G is missing. Therefore, you do not have any overlapping sequences. In both, G is missing, and then all the other downstream things are seen the same in both the alleles. So sequence analysis you do when you want to look for single nucleotide variations, insertions, deletions, and duplications of a few bases. And the molecular genetic tests that are used for sequence analysis are most commonly Sanger sequencing for smaller genes and specific mutations, and next generation sequencing for larger genes. And when you're looking at multiple genes or larger portions of the genome, such as the entire exome, all the coding portions of the genome, or the entire genome. So small deletion could be where you're looking at missing few nucleotides only. But if one or more exons are deleted or duplicated, they are referred to as large intragenic or exonic deletions and duplications. So these cannot usually be detected by Sanger sequencing, especially the heterozygous ones. And the next generation sequencing data can be analyzed for exonic deletions and duplications based on the coverage depth and the read depth. But the preferred modalities for testing for these large intragenic deletions and duplications are either multiplex ligation dependent probe amplification, that is the MLPA technique, or a high resolution exon level microarray. So examples of conditions which have large exonic deletions, the commonest ones would be spinal muscular atrophy and Duchenne muscular dystrophy. Now, this is a 17-year-old boy who was clinically diagnosed to have SMA type 3. And we know that 95 to 98% of SMA is due to deletion of exon 7 and 8 in the SMN1 gene. So therefore, here we would do MLPA of the SMN1 gene as the first line test. And that was what was done for this particular patient. And a homozygous deletion of exons 7 and 8 in the SMN1 gene was detected. You can see absence of the peak suggesting a homozygous deletion on both alleles. The exon 7 and exon 8 are missing. So that confirms a diagnosis of SMA3 in this patient. Now, another example of a large exonic variation, but in this case, a duplication. So here we have a 7-year-old male child who presented with proximal muscle weakness, very significantly elevated CPK, and also bilateral calf muscle pseudohypertrophy. The diagnosis here clinically is Duchenne muscular dystrophy. And we know that 65 to 70% of DMD is due to large exonic deletions and duplications in the DMD gene. So here again, MLPA would be the test of choice as the first line test. And we documented a hemizygous duplication of exons 44 to 50 in the DMD gene. Hemizygous because this is an X-linked gene. There's only one copy of this in this male individual, and that is showing the duplication. You can see the peak is more than what is supposed to be for normal individuals, and that confirms your diagnosis of DMD in this particular patient. Then moving on to other types of variants, trinucleotide expansions. So these are actually trinucleotide repeats are nothing but stretches of repeats of 3 nucleotides in some genomic regions. These could be in the intron, in the exon, in the 5' untranslated region, or in the 3' untranslated region. And when there is an expansion or increase in the number of repeats beyond a certain normal stable level, or which is also referred to as the threshold, then disease occurs. So these are some common examples of trinucleotide repeat disorders: Fragile X mental retardation syndrome, myotonic dystrophy, Huntington disease, spinocerebellar ataxia types 1, 2, 3, 6, and 7, Friedreich's ataxia, spinobulbar muscular atrophy. Of course, there are many others, but these are the relatively common examples. So for example, in this family with Huntington disease, now we know that the gene associated is HTT gene. Whoever has more than 35 repeats will manifest the disease. So these individuals, you can see the ones who are shaded, those are the affected individuals. All of them have one allele normal with normal number of repeats, but on the other allele, the number of CAG repeats is more than the threshold, and therefore they are manifesting the disease. And the tests for trinucleotide repeat expansions are usually not your conventional Sanger and NGS, but a different modality of testing is used, including triplet prime PCR and southern blot testing. Again, you will have a more detailed explanation of this in other webinar lectures. And when we talk about copy number variations, we are referring to a gain or loss of stretch of DNA, which is more than 1,000 nucleotides in size, that is more than 1 kilobase in size compared to the reference human genome. And the size of the CNV can range from 1 kilobase to several megabases. CNVs can again be benign or pathogenic depending upon the genomic region involved, the number of genes involved, and also the dosage sensitivity of genes within the deleted or duplicated region. And these are the tests which are used for copy number variations, including MLPA, FISH, and chromosomal microarray. These are some of the common microdeletion, microduplication syndromes. You have Wolff-Hirschhorn syndrome, Credeshart syndrome, then you have Williams syndrome, also Dye-George syndrome, and many others. Now, lastly, notation and classification of variants. I'll just quickly go through this. There would be a more detailed class on this later. So basically, nowadays, the Human Genome Variation Society guidelines, the HGVS notation guidelines, are followed for denoting variants at the DNA level, at the protein level, at the RNA level, and so on. And the classification method that is used for this is as per the ACMG standards and guidelines. So there is a set of guidelines given for classification of sequence variants, as well as a set of guidelines given for classification of copy number variants. And broadly, the variants are classified as pathogenic, likely pathogenic, variants of uncertain significance, likely benign variants, and benign variants. So that was all about variants and the structure of the gene and the genome. Thank you.